Hi, welcome to the lab, to the Glyxon Biolab here in Mexico City. I'm David Castillo. I'm the founder and director of Glyxon Biolabs. So today we're going to show you around our lab. So we're going to start here with our PCR machine, our real-time PCR machine next to the protocol bin and the pipette that we use for surface chemistry and the preparation of magnetic nanopit. And here we have one of our pets. It's a pirate, a biopirate, next to the tip bins, and another pet that is out there too because we work with plants well. And here our pipette and the PCR machine, the portable PCR machine, and pipette tips boxes, and a centrifuge, a vortex, and an orbital shaker. Here we are incubating a plasmid that is induced using light. And here we have our pH meter, a hot plate, dry bath, and another vortex. And like our sterile deionized water bottle said, biohacking is not a crime. Biohacking is science. Here's our sink. Don't look at it. It's full of dirty dishes like any other house. And here we have our reagent rack with all the reagents that we require for working with non-pathogenic enteric bacteria and tissue engineering in plants. And also we have our electrophoresis chambers for both DNA and proteins, and also material that we use for preparing Western blots like membranes and blockers. And we've got a blender now we need to liquefy something. Another pet here next to books. And we have our transilluminator, blue light transilluminator, another PCR machine, label maker, another incubator, Bunsen burner, liquid reagents. And here we have our power supplies, and for energizing our experiment for either protein or DNA electrophoresis. Next to the lab manuals, so here we have here the Sandbrook or the Maniatis and our protocols. Here we have our 3D printer that we normally use for 3D printing of microfluidic devices, and another incubator, a speaker because there must be music, and our microscope for the dissection of plant tissues. Here is the clean area, and I promise it's clean. And here where the magic occurred most of the time. And here we have material for maintaining the sterile conditions of the multiple strain that we work with, and also the multiple species of plant that we work in this area. And here we have the media and clean tubes and clean tips. Next to is incubator for petri dishes and a centrifuge. And here we have our Instapot that work as autoclave, a microwave, and our fridge. And this fridge we have here sushi and pieces of ancient brain. No, not really. Here we have reagents, liquid media, buffers, and everything that we need for the daily activities in the lab. Here's our rack for the plants. And here's some of the devices, open source devices that we use in the lab, including this Open Drop that we have used a lot in the past for the developing of protocols for the synthesis and the preparation of magnetic nanopit. It has been really, really helpful. And here's one of the protocols and process that we use for preparation of plants and for plant tissue culture. And here's ELISA preparation for antibody detection. And here is one of our colleagues. This is Wally Godinho. She's a member of the lab, and she's preparing some seeds in order to be propagators in specific media with hormones and inducers. So in the past, we have worked with several vegetable models, including potato, tomato, some coniferous, and tobacco plants as well. Among the techniques that we dominate in the lab are also protein chemistry, like electrophoresis and the preparation of recombinant proteins as well. And also the 3D printing using hydrogels infused with cells, the propagation of cell tissues, and the preparation and isolation of nucleic acid, both RNA and DNA, for the synthesis of our own plasmids and also the engineering of them. And also we're involved in conservation projects, like for example, this little guy that is a sequoia growing in our lab. And also in growing microalgae like Chlamydomonas reinhardti that is intended for the biofuel production. So we have been involved also with other groups, like for example, Ottawa Bioscience, that we were involved in the conception of a bioreactor, and also its implementation phases, like for example this one, that is the complete prototype that we were able to synthesize the enzyme that are required for the detection of COVID-19. We also been involved in several other projects for lab automation and robotics as well, with optics. And here's some of the institutions that we have worked in the past with. So please join us.